Good morning, sir. This is Sadiya Anzum from first BTech first semester from the Department of Computer Science and Engineering. My topic is potentiometric sensors and amperometric sensors. Potentiometric and amperometric sensors, understanding the fundamentals. Any sensors are the essential tools in many scientific fields. They use it to detect and measure the specific properties of substances. The two prominent and important types of sensors are potentiometric and amperometric sensors. These are called together electrochemical sensors. Let us see the working and the practical applications of potentiometric and amperometric sensors. First of all, what are the potentiometric sensors? So potentiometric sensors are a type of chemical sensors used to determine analytical concentration of certain compounds in a solution or glass. So let us look briefly working the potentiometric sensors. Potentiometric sensors are the sensors which measure the potential difference between the working electrode and the reference electrode is a potentiometric sensors. So here they are clearly mentioning that in potentiometric sensors, they use two electrodes. One is reference electrode and one is working electrode. So here a glass electrode is used as a working electrode. So glass electrode is a type of electrode which is ion selective electrode, and it only determines or identifies the H plus ions. Whatever the solutions, whatever the ions we take, they are present in the solution, it can't detect. It only detects the H plus ions of that solution. So it is an ion selective electrode which is used as a working electrode in the potentiometric sensors. So it detects the H plus ions and changes the potential of a working electrode, and the electric signals are displayed on potentiometer. Let us see the practical application of this potentiometric sensors. Applications of potentiometric sensors. Analysis of glucose in blood of diabetic patients. So the glucose, at what percentage the glucose is present in the diabetic patients is checked or measured by these potentiometric sensors. So glucose, it is a neutral. So it can't detect any electrical signals. So we change into it gluconic acid by oxidation. So it provides us hydroxide peroxide. So this hydroxide peroxide again releases the H plus ions, oxygen plus electrons. So the electrode, working electrode, which is a glass electrode, it is an ion selective electrode. So here there are the H plus ions. So it detects, it identifies the H plus ions and changes the potential of working electrode. So due to this change of potential in the working electrode, the electric signals and are displayed in the potentiometers. Means here we are checking and measuring the glucose in the blood. So due to this change of potential, the result means at what percentage the glucose is present in the blood is displayed on the potentiometer. So this is the practical applications. So we can see some general applications like potentiometric sensors are used to analyze drugs in biological samples such as blood, urine, etc. So these are also used in water and wastewater for cyanide and ammonia analyzations. And potentiometric sensors can be used in food processing also. So this is the working and application of potentiometric sensors. Next, we will move to amperometric sensors. What are amperometric sensors? These are the sensors which are self-contained integrated devices that measure the current resulting from the oxidation or reduction of an electroactive biological element. So these generally detect the current flowing from between two electrodes. So here also we are using two electrodes. Let us see the working and what are the electrodes we are using and what is the principle here we are using, and also practical applications. First of all, working of amperometric sensors. Amperometric sensors are sensitive analytical systems that measure current as a result of an electroactive substance losing or gain, oxidation or reduction, both redox reactions, an electron while undergoing an electrochemical reaction. So in amperometric sensor, we are also using two electrodes. It detects the current from one electrode to another electrode. Here also working electrode and reference electrode we are using. So the current is detected by losing or by oxidation or by gaining, like reduction. The electrons are released, and this is while undergoing an electrochemical reaction. We can detect the current between the two electrodes. So next point, a redox reaction occurs during the measurement that generates current. So a redox reactions, maybe the oxidation or reduction reaction is occurring during the process and which is generating the current in the form of flow of currents. So here also the current is identified by working electrode and generates the suitable electric signals on the displays. So here working electrode is identifying the current, and in potentiometric sensors it is identifying the H plus ions. Here it is identifying the current. Let us see the practical applications and understand the process. Here analysis of uric acid. Here we are taking uric acid in the place of glucose for any checkup of any patients or anything. So here also uric acid is a neutral. It can't detect any signal. So we are converting it into allantoin in the presence of urease, allantoin and hydrogen peroxide and carbon dioxide. So here also this hydrogen peroxide again releases H plus ions, oxygen and electrons. It is a redox reaction because here electrons are released. So in the redox reaction, one of the reactions, reduction or oxidation occurs. So here oxidation reaction occurred. So electrons are released. So due to flow of electrons, current is applicable. So these electrons are identified by working electrode. So these electrons move in the working electrodes, which changes the potential of the working electrode again, and the result. So due to this change, the potential of the electrode changes. So the results are displayed on the potentiometer. So this is the application, practical application of amperometric sensors. And it is the practical application. Now some other applications, general applications like amperometric sensors are used to detect medical analytes in blood or urine such as glucose, lactate, cholesterol, and like another environmental monitoring. Amperometric sensors can be used to monitor environmental markers like pesticides. In environmental monitoring also we are using amperometric sensors. In food quality monitoring also we are using amperometric sensors to detect the quality of the food. So this is the application and working of amperometric sensors. Now we will move to the considerations and comparison. Here potentiometric sensors and amperometric sensors are compared. Like these are high sensitive. Potentiometric sensors are high sensitive and amperometric sensors are real-time monitoring and fast response times. They selectively for specific ions and they are suitable for static measurements. Here amperometric sensors are suitable for dynamic measurements. So thank you, sir, for giving this opportunity.